So, it's my pleasure to introduce Scott Berry, Berry Consultants, to kick off this first of our simulation guided design webinars. Over to you, Scott.
Fantastic. Thank you, Tom. It is one of my favorite topics. Let me see if I can share appropriately here. Let me try this. And this. How does that look, Tom?
That's looking very good. I was all
okay. So one of my, I see next slides and all of that. So, okay, wonderful. So simulation guided trial design. I'm going to do this. I've got a number of examples to show you, and I've sort of hit and run and some warnings and tried to define simulation guided trial design. And in thinking about this, a little bit, and I'll tie it in a little bit of the history of what FACS is and how it exists and what other people think about it within it for this. And I was trying to think of the best way to define simulation guided trial design. And this probably misses the boat, but let me try this. So a man walks into a bar and says to his friend, "Oh, how's the wife?" And his friend says, "Compared to what?" And of course, the man that he asks the question to is a statistician. And if you ask a statistician, "How's your spouse?" the answer is compared to what? We continually compare things, and it's hard to evaluate something just in isolation. And I think this is really the crux of simulation guided trial design. It's why I think there's misunderstood. Simulation is one of the most misunderstood things within clinical trial science to some extent, because everybody thinks it's something different than what the person says it is or what they're using it for. And I'll talk a little bit about that, but really this is about the ability to compare different designs and say which design does better. And then you pick up components of it and say, "Oh, this is good, that's good, let's alter the design, let's go a different way." And it's this constant comparing is a critical part to this. So I'll sort of describe this. This is an actual Berry proposal. So we talked to a potential client. I've taken some stuff out, so you can't identify things here. But we talked to the client. They're interested in creating a design. They're interested in exploring innovation within the design. That can be in the analysis space. That can be adaptive design. It could be multiple components to it. And our proposal, as we lay out, okay, what are we going to do, is this process is at the heart of probably 60-70% of the proposals we provide for trial design kind of look like this, where, okay, let's take some candidate designs. Some of them fixed trials, some of them looking at adaptations, and we're going to simulate them. Step 2 is we simulate the trials in pretty good detail about enrollment rate, dropout rate. We simulate them. And what we're going to do with those simulation results is we're going to compare them. Step 3 says simulation results will be summarized and compared across the different design options scenarios. Initially, it might be sample size. It might be number of arms in stage 1 of the trial dropping for stage 2. And we compare the designs. Step 4 is iteration. You don't design an adaptive trial by throwing one up on the wall and saying, "Oh, that's it." Simulate it and put it in the protocol, and we're good. That's a weak use of simulation, and I'll sort of talk a little bit more about that. But really the role of this is to provide apples to apples comparison of more complicated designs. Which one's better? How well do they work? And so that's step 4. This iteration and repeated simulations are a natural part of a productive process of optimizing the trial design. That's what I think simulation guided clinical trial design is in that. When that process of then iterating is done, you write up the design, you create an adaptive design report within it that fully describes that design. So this is where things get a little bit, where people's view of this can be different, is they see an adaptive design report on that design. And this is part of the protocol, and this can be an appendix to the SAP. That doesn't typically go through all of the potential designs and show why it's a little better to do this than that, why we went this route, why we increased to 5 doses instead of 3 doses during this process of it. It talks about that design and gives us simulations for that. I think people seeing that document didn't see all of the 2 to 4 and the iteration, the simulated guided design. They saw the design. So I've presented on these FACS webinars previous trials of adaptive trials, showed what they look like and all of that. And you see that final finished product, but the design process was all of this 2 through 4 part of it. Will help you then write the regulatory documents, briefing documents, all of those pieces. So to me, this is simulation guided trial design. And this is how I'm involved, Barry's typically involved. This is a common process we go through for creating trial designs, creating adaptive trials, optimizing the trial designs. Okay, now I've restructured this. I've iterated this talk many times. I went through it. I always went through that process on this particular slides, and I had all my warnings at the end, and I moved them to the beginning. So I'll sort of do the warnings, and I'm going to show you some examples, and maybe I can kind of touch on those. Clinical trial simulation, when you talk about this, it means very different things to different people. It's partly why in that proposal we lay that out, because if we just say, well, we'll do trial simulation and all of that, the person reading that thinks that might be something very different than how we intend it to be used. For example, PK/PD scientists do simulation. They do a very different brand of simulation than simulation guided trial design. They're actually forecasting what's going to happen when we put this in the body, what's going to happen on clinical endpoints based on the amount of exposure and the amount of dose and various things like that. They're doing prediction. Many people think simulation is about prediction. If you go into common things that we see and read about it, every NFL season that starts, you'll see simulations of the season. Who's likely to win, who's likely to win MVP, all of these types of things. They're forecasting things. So people see this and, oh, it's a computer prediction of things. Now, you can use clinical trial simulations to predict a trial, but that's not simulation guided trial design, where it's being used as this complex tool to compare things apples to apples in this setting. Simulations, there's another hurricane out there somewhere in the Atlantic. I can't remember the name of it, but you can see computer simulations of the forecast of that. And people think of that and, okay, a computer's forecasting what's going to happen. So sometimes people come to us and they expect we're forecasting their trial, which we're not doing. In those cases, we're simulating the design under a range of scenarios, coming up with the optimal mousetrap to figure out the right answer in the trial. So people see this, they think of it very differently, and so it's a weird term. Now, even people involved in trial design use it differently, and it's used very differently at different stages of the design. In the adaptive design report, you'll see a summary of that single one design, and it's based on simulations. The guided design was this comparison of a bunch of different options coming up with the final design. That's used very differently. This in silico design process is different than presenting final operating characteristics for a protocol. Okay.
Scott, sorry to interrupt. Do you mind making your slides full screen with that use slideshow button in the top? We had a request. We see presenter view.
Okay. I'm going to blame Tom for not telling me that. How's that?
That looks good.
Awesome.
I thought that's what you wanted. Sorry.
Okay. So the other thing is, and I have to warn you of this, I'm assuming most of you are statisticians. We think like simulations. We've been trained on sample spaces and what's the distribution of X bar given theta, and what's that sampling distribution? To us, that's simulation. We don't have any problem thinking about simulating the roll of two six-sided dice and what that represents. It's kind of how we think. It's very, very natural for us. It is super awkward for other people. But for clinicians, for regulatory people, regulators, it's a different kind of way to think and understand things. And again, in the back of their mind, ooh, this is a computer kind of forecast thing, and I don't know if I trust it part of it. When to us, it's Monte Carlo integration. This is nothing other than a calculation thing. But other people see it very, very differently. You have to make the simulations approachable in their language and what they understand. So if you do a simulation, you just throw out their operating characteristics, they don't know what those mean. Even though they're trained on what power is and what type 1 error is, I'm not sure they know what it means. I did this once where somebody wanted to start looking at adaptive designs, and their initial design was 80% powered for a delta. And that delta would be a really nice drug. And they designed this trial 80% powered for that delta. I simulated trials and showed them the outcome of the trials, and 20% of those trials failed. And they were stunned by that. Wait a minute. If I have this really good drug, 20% of my trials fail. Now, we might chuckle at that, but that, by the way, that's our fault that person thinks that way, because to them, powering was just something you need in the trial. It's just something the trial you have to convince somebody that's a trial that's 80% powered. And by the way, you're a better statistician if you get a smaller sample size that's 80% powered. Wow, that's a good statistician. But didn't understand that was truly a risk with it. But when you simulate them and you show, okay, here's the outcome of the trial. This one meets statistical significance. This one does not. And you show them individual trials, and then you talk about what 20% is. Boy, they get it. I mean, because that's their language. They understand the final outcome and all of that. It's incredibly approachable for them, where generally operating characteristics are really weird. But to us, oh, that's our language. It's exactly what we think, but you have to work up to it. Now, after 2 or 3 calls, they know what it is. And you say, okay, here's one trial that's not statistically significant. This one is. The sample size was 528. And all. Then you say, that was one trial. Now we make this assumption and we take the average of 1,000, and here's what that looks like. Oh, sure, that's great. That's natural. And they know what it is in those settings. So this is a weird language for a lot of people. And it's hard for statisticians because it's so natural. We just throw stuff on there, talking to another statistician, everybody gets it. We're talking a foreign language until you get them on board. It's minimal usefulness as a final task. in that. And so if somebody comes to us and say, "Oh no, we know the design. We have the design. We just want you to simulate it." That's not interesting for us. We're not designing anything. We're just making a calculation. Now, it doesn't mean we don't do this, but it's not a great place to be. So I'm going to show you an example of one that we agreed to do it in part because when they described the design, I thought it was a dumb design. Now, I thought it was a dumb design because I thought there were better, more efficient designs that I thought that they would want to do. Not because I thought it was dumb, I just didn't think, in their goals of what I understood, I didn't think the design met them. So if I simulate this trial and say, okay, here's your design, here's what it looks like, they're going to say, oh, wait a minute. And that's in part this idea that they can write stuff down, and if they're talking statistics and power this and do that, and we want to do this, all of a sudden when they see it back in their language and they see it, it's, ooh, I didn't really want to do that. And that's hard for non-statistical people to do, is to envision, when I put down a flow chart, what does a data set at time 3 mean? What does this look like and that look like? It's easy for us, but it's really hard for them. Okay, so here was an example of one of those trials where I didn't think it met their goals. They were doing a Project Optimus type trial design where they were going to enroll the control, the low dose, and the high dose, enroll 70 per arm, and then they were going to pause enrollment and wait for 96 events, progression-free survival events. They were then going to calculate the one-sided p-value individually for each of the 2 doses. If both there are above 0.33 on the log rank test, they're going to trigger futility, and they're not going to go further, and it's done. If at least one of them is less than 0.33 p-value, they're going to pick a dose and they're going to go forward, and they're going to pick the smaller p-value. That's their trial design, and then they would enroll that target selected dose if they go to stage 2, and they would enroll another 100 and, I can't remember, 170 more per arm of those arms. And then they would wait for 201 total events in stage 2. By the way, there's an awkwardness to that, because does that count the events on the arm not selected, for example? And how does that affect power and all of that? Sometimes simulation brings out awkwardness in the definition. If you can't simulate it, and I don't know when 201 happens, you haven't defined your design completely. So that needs to be sort of, set up. And then, by the way, if you ignore that one arm, somebody needs to be unblinded to all that to tell when that happens. And is that okay? But then the final analysis for for progression-free survival, and they had set up the alpha level that they were doing 0.005 on progression and 0.02 on overall survival. They were splitting alpha that way. They wanted to be able to win one and not the other. So they were splitting alpha on these analyses. And that was their design. Just we need you to simulate it and put the operating characteristics in the protocol. So I simulated it in this, and here's 15 different truths. We're not going to jump into too much of this, but I walk them through. Here's 15 different truths. By the way, I show them example trials. I'm going to show a couple to you. But here's then a summary of that, and they're really targeting 0.7 as a hazard ratio for progression-free survival within that. Now, one of the doses might be 0.9 and one's 0.7, and they want to pick the 0.7 dose and then move to stage 2. So here's a number of potential scenarios, and of course 1.1 is the null hypothesis. And then the likelihood of stopping for futility. You can see that column for stopping for futility given the scenario. So under the null, 50% of the time, both p-values are above 0.33. Within that, the probability of advancing in which dose is selected. So if you go forward with 0.9 and 0.7 as the two doses, 13% chance of futility, 12% chance you go forward with the low dose, which is 0.9, and 0.73 you go forward with the high dose. And then I showed them some example trials. Here's an example of when you have your 70 events and showing them the control arm, the low dose, and the high dose. So here's an example where at this particular time, and you have the exposure and you have your dropouts, here's what it might look like where the hazard ratio is 0.94 for the low dose, 0.312, and I'm showing them their design. This is probably the first thing I would show them. Here's an example of your design. Here in this design, you pick the high dose. You go forward to stage 2 and you do the analysis. And now you still have the data on the low dose, but you didn't enroll anymore. But the primary analysis is the control against the high dose. And in this example, the final hazard ratio of that's 0.629. And the p-value is essentially 0. This is a successful trial when you get to 201 events. And so I show them an example of that. Here's an example where you might stop. You don't continue forward. And then I actually had a slide of like 100 examples and walk them through a number of examples in that. By the way, I don't tell them the truth of what was the scenario that generated it. Based on my numbering system, you could probably figure it out. Well, what the truth was that, but would you be happy with this decision? The statistical stuff you threw in the protocol, is this a good decision in those scenarios? And then when they kind of understand exactly what the trial looks like, I might show them, so here are the p-values at the interim. And the different p-values, and the green ones you go forward. Blue ones you do not. And it's pretty straightforward graph. If both of them are above 0.33, you don't go forward. If one of them's less. So you could have a p-value of 0.3 on the low dose and 0.8 on the high dose, and you're going to go forward. Here are the hazard ratios that would be associated with those same p-values. So if you saw a hazard ratio of 0.8 on one arm and 1.2 on the other arm, you're going to stage 2. Is that a good decision? Now, what this brings out is all of a sudden, analyzing these independently might not be the right decision. I mean, we kind of think the effect of both doses are pretty similar in that, maybe it's a little bit tolerability, safety. That's a huge thing to understand what they envision these two doses to be. How is it in context? And all of a sudden, maybe I don't want to go here, but maybe here I do want to go kind of thing, because now they're both somewhat similar. I have better trust in the hazard ratio. So things along this line, but maybe I want to do Bayesian borrowing. Maybe I want to do a dose response model that I wouldn't go here, but I would go here. And now all of a sudden at the interim, this is the rule you set out. And then we walk through the various operating characteristics of this. And interestingly, one of the things that they pushed back on was their original alpha thing. That if it's 0.7, the probability they win the trial with that is 72%. Because of the way they did this progression-free survival, had you spend full alpha on that, it's 85%. So I'm already showing them alternatives to their design in a way, just because I just want to show you, what's doing here, and I'm showing them this. Now, in the back of my mind, I know there's things awkward about this, and walk through this, Now, what happened at the end of this? Oh, the other thing I showed them, and it's one of the really nice things about simulation, I can calculate some pretty detailed things, is I show them how long their delay is going to be from done with enrollment to observing 70 events. So I presented the interquartile range of the delay in weeks under the different scenarios, and they think they're down here in this sort of 0.7 type scenario. And, this is a 6-month delay. And they weren't super comfortable with that. And then what are we going to do at sites and all of that? So they start asking, what if we keep enrolling, but we do this decision at 70 events? And now they want to look at another simulation. Okay, what if we do hierarchical testing, that we do PFS and then OS in it? And invariably, when you test any design, so even in the circumstance that they knew the design, it changed. By simulating it and showing it and contrasting several things and showing them data at the interim of what it did, not super comfortable with this thing we built. And it enables you to put things on the language they understand, the data understand the parts of it, to really vet the design and then contrast it. And then the next thing they wanted is, okay, let's fix this design, but let's look at version 2, version 3, version 4. And I was just looking at my directory for this simulation, and I had simulation 1, simulation 2, simulation 3, and those were iterations of the design where I was doing the new simulation. So even in a scenario where the goal wasn't this simulation guided, simulations brought that out, and that's sort of what happened by vetting it, and I showed them comparisons of things they didn't necessarily do. So, I'm sorry, the expectation on that, and it was the design iterated within that. The other thing that I sort of hear in this, and early on when we started doing this, clients would almost ask for this. Okay, you're starting on this. Do you have a simulation plan? Can we write out a simulation plan? And I think it's counterproductive. And it's almost like we jump in and in meta-analyses, we write down everything we're going to do. In a SAP, we write down everything we're going to do. This is different because this is exploration. And immediately upon that first set of simulations, several things happen. First of all, you really dive in and you figure out what the goals are, what operating characteristics are important, what performance things are important. So you iterate on those, and then invariably by looking at the simulations, you iterate the designs you want to explore. You can't pre-plan all of that. It's counterproductive to do that. It limits the exploration you do during the simulation guided part of it. So I know this is something we do a lot as we write down plans and all of that. I think it's counterproductive in the design. Now, in some cases where you walk in, you say, okay, I have a design and I want to present operating characteristics. I want to demonstrate control of type 1 error of my design. That might be a place where you write in, here's the seed I'm going to use, here's the number of simulations. And that could be a place where that's done at the very, very end of this process. But it's counterproductive early. It's not simulation guided design. It also, if you try to envision all the possible designs you go into with 3 arms, 4 arms, 2 arms, changing when I make the decision about the dose, the sample size, the adaptive part in part 2, you're going to spend 2 months doing the simulations. You're going to throw it on everybody, and they're not going to know what to do with it. It's massive. And almost invariably when you do that, all of a sudden the game changes. Oh, okay, I wasn't really interested in that. And you spent 2 months on simulations and this huge plan, and all of a sudden everything's different once they start looking at results. Okay, so it's very much this design process where you start with designs, you do simulations, you evaluate them, you compare them, you come out with new designs, you iterate them, you repeat this process, you get a final design. And then within that, now you're going to validate that code. You're going to do final simulations and adaptive design report. You're going to verify those simulations. You're going to package that code in a way that people can verify. You might write a simulation plan here when you're done iterating, but you don't want to do it before you're done iterating. Okay. So when you're doing simulation guided trial design, you'll spend as much time creating the comparisons of the designs as you do running the simulations. So your activities in that, I have to write code, I have to write the simulations, I've got to go in to figure out how to make facts work within this. I've done the okay, good. I've simulated the 3 designs. Now you've got to compare them in ways that allows your team to make decisions, to move forward, to understand the designs and the different components of it. You're going to spend more time on that part than you probably spent on the simulation part. And so it's sort of envisioning, how do I want to show this? How do I want to show example trials? For example, in the case of that decision process at 70 events, I didn't think it was enough to say, here's a table of the hazard ratio and the p-value. I wanted to show them the data. You're going to see a Kaplan-Meier plot like this, or the people making the decision. And I wanted to see, so they understood it deeper, what they were doing, and then the p-value and the components to it. I thought that was an important part to understand the design. So in the first round of simulations, I want to make sure they understand the design. Otherwise, when it's not working great or other signs are different, how are they going to know what to fix if they don't really understand the machine? So I'm going to spend a lot of time on example trials, and we spend a lot of time early on example trials. Then once you get going, they understand operating characteristics. It's much easier to be comparing them. And a lot of times, I ask myself, what would I do? If I were making the decision, what would I do? And I look at my material, and usually I don't have the right graphs for that. I'm going to graph 3, and then I'm going to a different page to compare a different design on that same 3. And I say, you know what? I got to put these on the same graph. They've got to see what this looks like in comparing the designs on that. And then I put it, yeah, I'd pick this design. Actually, I'd alter it a little bit and I'd go forward, but I really like this. And then I make sure I'm presenting that in ways that I understood how to make the decision and all that. And okay, every once in a while you'll be asked, what would you do? As part of that. And then I can explain it. So I'm thinking about the graphs, and you're going to spend a ton of time on that part. Identifying the goals of this are really hard. And teams may be programmed in how they talk to the statistician to talk in statistical things. Oh, we want the following power, and proof of concept in phase 2 is statistical significance. In part, that's what they've seen before and all of that. Rarely is that true, actually. It doesn't really talk about, am I going to be successful in phase 3? Can I convince somebody to invest in this drug? Is it meeting a target profile? Rarely are those tied to statistical significance. It all depends on the size of the trial and the number of arms and the components to it. This is really hard. But it's the huge value of simulation to show them exactly how this fits in development, how my selection of the dose in phase 2 affects my likelihood of success in phase 3, and getting that all right. So I'm talking their language, development timelines, development success, the proof of concept. What is that? And that's the really hard part here. You got to be part of these discussions and all of that. It doesn't work that they talk about the science of it all and they come to you and say, we want to see X, Y, and Z. Because you're not going to be able to do that part well. You're not going to know what to present. And when you ask yourself, what would I do? Well, I don't really know. How much do biomarkers play a role here? How much does this play a role? What is it I'm trying to accomplish? Why am I doing dose finding? Is it to satisfy regulators? Is it to satisfy making sure we have the right dose? Is it about safety or efficacy? All these things need to be taken in to present the right performance parts. And that's really, really hard. That's something you iterate as part of this as well. You won't have all the right things the first time. They'll say, oh, wait, can you show me X or can you show me Y within that? And you may not have it, but the next round you're able to do that. And if somebody comes and says, oh, here's our synopsis, design an adaptive trial, it's sort of like, that's the old thing of how's your spouse? Well, compared to what? I don't know. Every design's a good design, but does this meet your goals and all that? That's a really hard part of it. And that's what takes time to do it. I got this email last week, and I've altered it a little bit to make sure nothing confidential is here, but this is almost verbatim the email. And they want to send a design, and that doesn't. I need much more than that. Hence, let's discuss this. And we talked about it Monday, but for a dose finding, and so they're sort of giving me the design for a dose finding and sample size re-estimation, seamless 2-3 trial design. They want 2 doses, 1x and 2x, and their dose dropping options. If the Emax model does not apply, select the low dose. If Emax model cannot be refuted, select the highest safe dose. Or use linear regression change from baseline after inclusion of approximately 30 patients to select the most effective dose. And then sample size re-estimation based on the primary endpoint after 30 patients have completed 36 to 40 weeks of follow-up. So tons to unpack here within this, and they've given me statistical things. No, I don't think they're applied well. I'm trying to identify what is the problem you're trying to solve. Why are you doing the Emax? What do you mean by that? And what are the endpoints they're putting in this? And interesting, like he said, if it cannot be refuted, select the highest safe dose. So they want to do this sort of combination. And saying the Emax model doesn't apply is awkward because a flat line is an Emax model, a decrease in, but I don't, I mean, this is not to be critical in any way of them misapplying a procedure. I'm trying to identify now, what are the goals of this? What are they trying to do? And really, why they do dose selection? And I think it's probably a little bit of efficacy and safety both. And then they say, they use the dose groups combined versus placebo for sample size re-estimation. So, they probably think these doses are similar in efficacy. And I know a little bit about it going into this, but these are all the questions now I'm asking them. I'm not going to go off and do simulations yet. I'm not going to simulate that design for a couple of reasons. The next slide, I'll talk about that. But this is giving me a light into what are the problem they're trying to solve, that now I can start to lead to good solutions to this. And then we have a call where I get to ask my questions. And I don't say, what are the 3 parameters? Do you want a 3-parameter or 4-parameter Emax model? I'm really trying to figure out what problem are you trying to solve. And then I might say, suppose at the interim you saw data X and Y. What would you do? And now what happens is they start talking out of statistical mode, and they start talking about in the drug development side of things as to, this is what I would think, and I would look at tolerability, and if there's no safety issues, I'd pick the higher one. Or, even in that circumstance, I'd pick the lower. And I start to give them example data sets, hear what their goals are and what they're trying to do, and then let me figure out the right statistical things to do in that, rather than them putting their, and maybe an Emax model is the right way in that setting. So have those discussions. The other part of it is it wouldn't be a productive thing to simulate that design. It's not completely specified, so I don't even know if I could, but if I tried to do that, because almost immediately if I presented that design, the whole world changes. The first scenario you show them, it's, oh, well, no, I mean, I don't want to do that, and I don't want to do this, and can you show me this? And now I've spent 3 weeks simulating a design that it's really just a get started thing. So start simple, show a fixed design. And by the way, that was Monday, and now I'm charged with what's the first steps. I'm going to show a design where they carry 2 doses all the way to the end. And they use split alpha. And that sort of brackets things over. You could do this. You could do at 30, you pick a dose. At 50, you pick a dose. And they're fixed. Otherwise, it's fixed, relatively simple. And I'm going to create a rule for that. And by the way, you saw a really important part to that is the endpoint is at 36 or 40 weeks. In the back of my mind, I'm thinking, how am I going to use patients that are incomplete? When I make that decision at 30 patients at 40 weeks, I know a whole bunch more in that trial, and it's informative, and it's hard for you because you don't know the disease, but it has to be informative in that scenario. So now I'm avoiding that in the first simulation, and I'm showing them relatively simple. I can turn this around in a week and show them the first round of this, and the whole world changes in the first one. And I learn a whole bunch what they're thinking and all that, and then I can add features on. Okay, here's what we do here, here's what we do here, and we understand the effect of all of that is typically the approach to that. Okay, let me skip this one, timing-wise. Those were some graphs I showed last night, 5:30 my time I showed that. Here's an example of last week where there's historical data, there's a new trial, and it was looking at, based on the current trial, here's success if you do no borrowing of the old data. And whatever the old data is, whether this is pediatric to adults or whatever the scenario. But if you do no borrowing, would be successful in that trial. Typical 0.025 trial. If we do 0.1 borrowing of the other data, these trials turn successful, and we're showing them if you pool the data with that, these then turn successful. And here's your probability of winning the trial under the null. And here's the probability in the different scenarios. Here's the characteristics. These are things we're showing them in comparisons. That's comparison of 11 designs. 11 approaches to the analysis. It's all about comparison and understanding what it is and what happens in the design. And a lot went into thinking about this particular presentation of the material. I'll show this one just as a two-dimensional population finding example. And showing, if the effect is in the upper left, where the thresholds and 80% chance that we pick that population, where you think a lot of times enrichment are two A and B, or 7 groups. Here was a two-dimensional plot of trying to figure out the right population for something. And we've got a model in there. We've got adaptive designs. And these were the graphs we were showing to try to show the contours of that. Liz Lorenzi created these graphs, and they're fantastic in that. By the way, Jessica Overby created these graphs. So I'm using their work in this. Okay. The other part of this is, and this is a little bit of my initial joke about the spouse and compared to what, the value of simulation guided design is, the proof of it is the existence of the promising zone design. And the promising zone design by itself, how is it? Seems reasonable and all of that. But once you start to compare it to other designs, it's almost never better. I've never seen it perform better than alternative designs. So if you simulate the design and you present, here's the promising zone, cool, nice. Compare it to any other design and you say, oh, doesn't work very well. And so here's an example of a comparison of a promising zone design and how do we do adaptive phase 3 trial. So very straightforward. Standard deviation is 12. We have effect sizes from 1 to 6 or 0. Higher's better. Very generic scenario. So my potential effects of the treatment, 1-to-1 randomized. The team's contemplating a design of 300. If they run a fixed trial, they're contemplating 300. By the way, if somebody says, what sample size do I need for an effect size of 4, saying 300, I feel like isn't really the simulation guided thing. It's presenting this. So this would be my response that, yeah, with the effect of 4, I'm 82% powered for a sample size of 300. But now I see, by the way, if my effect is 3, my power is 0.6. You don't get that if the answer is 300. And, what do I need to be powered on that kind of thing? So you get to envision and you're comparing designs. This is comparing designs where the thing I'm trying to figure out is what's my sample size? And, I could run a sample size of 500 and I'd be powered. 3 is a marketable drug. If the effect is 3 and I'm underpowered for that, but 500 would be 80% powered. If I need to go there, I'd be willing to go there. There's a lot of times where people, let's do a promising zone in this case. And 300 if we can. And if we need, let's go to 500 and to catch the 3 effect. But the 4 effect, 80% powered for that. So common promising zone design, which I'm assuming most of you know this design. It's a really neat, clever trick to the promising zone design, which is part of its appeal is I don't spend alpha. So you can write down a potential promising zone design where at 200 patients, I'm going to do an interim. And at that interim, I'm going to decide, do I go to 300 or do I go to 500? And the promising zone almost always throws out a 0.001 at that interim for already winning. I'm assuming immediate information on patients. So if I have 0.001 level, I'm going to stop at 200. Now, the neat trick about the conditional power is a bit of the negative of the design, is that the lower bound of when I'm allowed to go to 500 is you have to solve a little integral, and here it's about 0.4, where I can go to 500 and I don't adjust alpha. It's a beautiful mathematical trick to it. But if it's less than 0.44, I have to stick with the original 300 in the design. So that's the design in it. And the magic is that when I get to 300 or 500, I have 0.025 or 0.0249 because I spent 0.001 at 200 for potential early success. So here's the promising zone design. And it's a presentation of it where it shows, the top shows the effect sizes, 0. So I've simulated 1,000 trials from 0, 1,000 from 1, 1,000 from 2. Down here are the effect size of the final results. And what I've done here at 200, if you're above 5, you win in that at 200 and you hit the 0.001. So it shows them what effect size is right at 200. I'm going to declare superiority and the trial's over. And then those dark green are up here under each scenario. So if the effect is really 6, I've got a 60% chance of hitting that. Now I've moved these to the left over here because they're trials that continue on. But I'm showing the effect size and the eventual result of them. These all in this scenario of what happens in that, as the trials that either go to the original 300, and I'll ignore this lump for a minute, but if they go to 300 and stick with that sample size, you're going to win the trial if you're above 3. the observed value above 3. You're going to lose if it's below. If you get out here to the end of the trial, you're going to win if it's above about a little over 2.5. And that's the value of the 500 to be powered at 3 is winning at about 2.3, something like that. Now I've colored these by you win the trial if it's green under those scenario. You lose the trial if it's pink. If it's yellow, you turn what would have been a loss. Sorry, I'm comparing this to a fixed 300 patient trial. So the first thing I'm going to show you what happens if you just run a 300 patient trial. That was the original trial. Now the promising zone, the yellow ones in that scenario is moved a loss to a win. So had you run the 300 yellow, you would have lost, but the promising zone makes that a winner. Those are great in the design in that. A blue one here is a win on the 300, moves to a loss. So some of these you would have won at 300, but because you went to 500, you ended up losing. And that happens with some probability. That's purple. That's kind of bad in that scenario. And the purple here is the promising zone. You hit the promising zone and you end up losing the trial in that. And now you can compare this to the 300 patient design. If my effect is 4, my power was 81%, and we saw that. Now my power goes up to about 86%. My sample size is 342, and my sample size was 300. So I've bought with 42 on average, I've increased my power in that. What's interesting is that power, I get almost the same if I run a 342 patient trial. So if I now change this to 342, I get power that's 86% in that scenario. And now the boost I got here over here in 3 is a little bit of yellow, a little bit of purple, which is bad. And my overall power here is a really small boost in this scenario. And largely, you can see that what happens is the trials that go forward were likely to win anyway. And you've now increased the sample size and the number of trials that would have won at 300, and now you go to 500 and you win anyway in those. So a lot of these scenarios, the green ones, you would have won anyway. A win to a win, and you increase your sample size 200. Okay, so that's great. So let's compare to a group sequential design at 200, 300, 400, or 500. Let's spend alpha in that scenario, and let's look at a group sequential design. So now when I do the comparison of the group sequential design to the promising zone design, in that scenario 4, where I was 80% powered at 300, my average sample size is 310, and my power is 96%, as opposed to 86%, and my sample size is smaller. In the thing I was worried about, the 3, in that scenario, my power jumps up to almost 80%, and the promising zone's at like 62%. And, my average sample size is not much different in that scenario. And so I can evaluate and compare two designs. You'd be hard pressed to pick the bottom design here in terms of its behavior. It uses more resources in those scenarios. And notice out here in these scenarios, it's using more resources. And I'm not getting much for that in the scenario in comparison. And by the way, you can move forward and you can envision futility rules now going in there, evaluating futility rules. The cool thing about visualizing this is at 200, by the way, the group sequential design, it's a promising zone. That zone is here. We want to go bigger. And then we have another one. Are we there yet? No, we go to the next one. It's actually what a lot of people bring the promising zone in to do, but it doesn't do very well because it's dictated by conditional power has to be in that range. It does actually function very well. And once you compare it to other designs, you realize that actually it doesn't do what I want as the director, what I'm trying to do, what I'm trying to optimize. And all of a sudden, this design does exactly what I want, but doesn't have the fancy name that promising zone. You can evaluate what's the cost of doing interims and all of the various things. It puts it all in a thing that you can evaluate apples to apples, scenario to scenario. And the promising zone rarely falls out on top of that scenario. Okay, I'll skip these in it. So the opportunity here is that trial simulations allow you to speak everybody else's language in terms of the likelihood of success, the average resources, the length of time of the trial, the probability that we're successful after selecting the dose, the impact that that selection has. And now you're starting to have development decisions within that tied to the things of their interest. And you're not having them awkwardly talk about power or specify things. And you get to evaluate which works better in their language. It's a really powerful way to use simulation within that. And I think that, to me at least, is simulation-guided trial design. Tom, turn it back to you.
Great talk, Scott. Thank you. Yeah, really nice with those current examples and reprise of the promising zone demolition. Yeah, thank you very much. I don't see any new questions. So yeah, thank you. Oh, we have one here. I did get, all right, someone just agreeing the promising zone is not better than the group sequential design. We have had a request. Will you be able to make the slides available?
Sure. But
Okay.
I'll PDF them so you can't move the boxes, for example, and see things. And I might remove a graph here or there. By the way, a beautiful thing about the promising zone to group sequential is now all of a sudden, suppose my endpoint is 40 weeks. And maybe I have incomplete information when I'm making those decisions. Hugely valuable to be able to simulate that part of it to pick the right design. And usually it ends up that way because rarely do we have complete information on them. Yeah, happy to make the slides available.
Thank you. Okay, and just to remind everyone that the webinar series is going to continue next week, where we start looking at, we're going to do things in order. So we start with phase 1, and we'll have a couple this week and next week and the week after, we'll be looking at different use of simulation in different dose escalation designs. So thank you, Scott.
Thanks, Tom. And thanks everybody for tuning in.